Good morning, and thank you to the organizer, to Mitocon, for giving me the possibility to present this talk. Today I'm gonna summarize the state of the art and the future perspectives about Leigh syndrome. So Leigh syndrome is a rare neurometabolic disease in childhood that has its name by Archibald Dennis Leigh, who was a British neuropsychiatrist who first described the condition in 1951. This is a severe disorder, a progressive disorder that usually has its onset in infancy within the 2 years of age. And the prevalence of Leigh syndrome has been estimated to be about 40,000 live births. The condition is more common in some population. This includes the regions of Quebec and Canada and the regions of the Faroe Islands. Leigh syndrome is defined by a typical neuroimaging pattern, as you can see in this slide. Typical are lesions in the basal ganglia and in the brain stem with necrotic aspects. This slide is to let you see that mitochondrial diseases have a great genetic heterogeneity, and Leigh syndrome can be caused by many genes. Mitochondrial genes that cause Leigh syndrome can cause other mitochondrial disease with either phenotypes. So in these slides you can see as mitochondrial genes like ND3 or ND5 can give a Melas syndrome or a Leigh syndrome. So many genes can cause Leigh syndrome, and these genes can cause defect in these genes can cause different biochemical defects. So complex 1 deficiency or complex 2, complex 3, complex 4 deficiency. So typical, quite common is Leigh syndrome caused by complex 1 defect. So quite often Leigh syndrome is associated with a complex 1 defect caused by mutation in complex 1 genes. Other genes involved in different biological mechanisms can cause a Leigh syndrome, and you can see here lipid and cofactor biosynthesis or genes involved in mtDNA maintenance like POL-G or SUCLA2 and others. Leigh syndrome has a genetic heterogeneity, but also in the context of Leigh syndrome we can have many signs and symptoms that can help us to define the diagnosis and to orientate the genetic algorithm, the genetic diagnosis algorithm. So we can say that some genes involved in the amino acid metabolism like HIBCH and ECHS1 can give an abnormal plasma cyl-carnitine profile and also abnormal urinary organic acids that can help us in make the diagnosis, or SURF1 that is an assembly factor complex 4 usually give a Leigh syndrome with an important myopathy and hypertrichosis and so on. So we have some diagnostic clues, clinical clues that can help the clinician to define the genetic cause of Leigh syndrome. And in this scenario, important are the profile of urinary organic acids that in some condition are essential to get the right diagnosis. So we have in Leigh syndrome, in some forms and of Leigh syndrome, we can have a trimethyl-glutaconic aciduria, a mild methylmalonic aciduria, like in defect of SUCLE2, alpha-2-ketoglutaric aciduria in defects of PDH. So this is an important urinary organic acids are an important marker to orientate the diagnosis. Also, as we have an important genetic and clinical heterogeneity in Leigh syndrome, we can say that different genes are associated with different outcomes. So mutations in MTFMT that cause Leigh syndrome have been reported to be associated with a better prognosis. Mutation in other genes that cause Leigh syndrome are associated with a severe, more severe prognosis. In our experience, we also faced a diagnostic challenge to define the cause of Leigh syndrome in a cohort of our patients who have a relatively mild form and quite stable in time of Leigh syndrome caused by compounded heterozygous mutations in DUFA F6, which is a complex factor, assembly factor complex 1. In these patients, we found a missense variance associated with a deep intronic variant. This is a multicenter study on Leigh syndrome, and as you can see, focused on predictors of survival, and is the main result of the study is to highlight as an early onset. Earlier the onset is worse is the prognosis, and patients who have an early involvement of brainstem at MRI are usually associated with the worst and worse prognosis. This is also confirmed by an Italian study, who is now submitted for publication based on the Italian cohort of mitochondrial patients with Leigh syndrome. Also in our study, we found that an early onset with an early involvement of brainstem is associated with a worse prognosis, and also we found that the most common form of Leigh syndrome in our cohort is the complex 4 Leigh syndrome associated with complex 4 defect by mutation in SURF1. Some months ago, we participated and organized and then participated to the Leigh syndrome ENMC workshop, and the main goal of the workshop was to gain consensus worldwide on the frequency distribution of the different genetic forms of Leigh syndrome, and also to give importance to developing of outcome measures for future clinical trials. So participants were from all over the world. Here you can see the names of the participants. The purpose, as I said, of the workshop was to be prepared for future clinical trials in Leigh syndrome, to gather information on the genetic epidemiology of Leigh syndrome, because if we know more about epidemiology of Leigh syndrome, we can also gather more information about natural history study in syndrome. That is very important, as we will see later on, to reach consensus on primary and secondary outcome measures. This is very important for the developing of clinical trials, and explore the possibility of biomarkers for the future clinical trials. The Leigh syndrome roadmap project is an international project that is focused on Leigh syndrome as mitochondrial condition for which invest funds to fund research project for identifying biomarkers and identify outcome measures for future clinical trials. So these are the focuses on Leigh syndrome roadmap project. So to improve the diagnosis of Leigh syndrome, to identify possible therapeutic strategies for Leigh syndrome, and to optimize the patient care, and also uniformize the patient care all over the world. So patient groups around the world have partnered to raise funds in support of this project. Why this was not done before? Because it was difficult choosing a subset of disease to focus upon in the field of mitochondrial diseases, for the poor understanding of preclinical models to begin with therapies, and for the historical lack of a strong network between the different centers that are involved in the studying the cure of mitochondrial disorders. The main goals of the project in the next 3 years is to develop, as I said, natural history studies that are essential to start to understand the proper outcome measures for future clinical trials, and to identify the best preclinical models and biomarkers to study upon. Why the natural history studies are so important? Because we have to define the current state of knowledge. We have to know which is the impact of early diagnosis in this severe condition, which is the impact of an early intervention in this condition. And also it will be important to know which is the real benefit of the mitochondrial cocktail in the treatment of this condition. It is usually in the clinical practice all over the world. And if our knowledge of the natural history, can impact in the development of therapies for Leigh syndrome. Mitochondrial disorders are rare. As I said, are clinically, biochemically, and genetically very heterogeneous, and also are unpredictable. So the disease course can vary in the same genetic group within the same family. The disease course is unpredictable. Are multisystemic disease with involvement of different organs, and patients are often very disabled to perform functional tests. So it was difficult, it has been difficult to define the right outcome measure to use, or the right test and outcome measure to use in these patients. In this article, in this paper, has been developed an algorithm to identify the right functional test that can be used in mitochondrial patients. This is highlighting what I was saying. So in this paper, 4 subdomains were identified. Domain 1 that was focused on intellectual function, energy and drive, psychomotor development. Domain 2 more focused on the structure of the central nervous system. Domain 3 was more a functional domain, and domain 4 was about parental stress. Another workshop was held in 2016 to identify outcome measure to be ready for clinical trials in primary mitochondrial myopathies. It was held in Rome. Primary mitochondrial myopathies are criteria to define a primary mitochondrial myopathy. What was identified was reached a consensus by different experts to set the criteria to diagnose a primary mitochondrial myopathy, and then the meeting was focused on identifying the best outcome measures for clinical trials in these conditions. This is the paper that was produced after the meeting, and as you can see, were identified tests for adulthood and childhood in clinical trials, and the 6-minute walk test has been identified as a very good measure in primary mitochondrial myopathies. Also, the consensus was obtained about biomarkers, and you can see GDF-15 and FGF-21 reached the highest consensus as biomarker to be monitored during the clinical trial in mitochondrial diseases, and also metabolomic studies. In the Leigh syndrome roadmap project, there is a first study that is going to be activated, is an early to start about as a natural history study in Leigh syndrome, and these are the center and the countries that are involved in this study. And these are the clinical scales that we will use in this natural history study. I thank you for your attention.
Hello, my name is Alessandro Prigione from Heinrich-Heine-University in Germany. And today I'm going to present our work that we're doing on mitochondrial disease, Leigh syndrome, using induced pluripotent stem cells. So I'm going to share the screen. Yeah. So so basically we use stem cell models for understanding Leigh syndrome and for also potentially finding therapies. And so with these models, one can generate neuronal cells in 2 dimensions, or also in so-called brain organoids. This is this picture represented here. And these are 3-dimensional organization of cells that can sort of resemble some of the feature of the developing brain. You see some cellular organoids in sort of something resembling of the human brain ventricles, and progenitor cells, neuronal cells, glial cells organized in a structure. So I'm going to start by asking a question: why there are no therapies for Leigh syndrome? And we know it's a complex disease, can be caused by nuclear mutation, here represented in blue, for example, mutation encoding for genes, mutation in genes encoding for proteins, of the complex 1, 2, 3, 4 nuclear gene, or mitochondrial genes, complex 1 and 5, for example. But still, this could be also good news because it means that we know the cause of the disease. There are mutations in nuclear mitochondrial genes, so that's a very good start to understand the disease mechanism. But unfortunately, in order to really understand the mechanism, we need a model system so that where we can investigate and understand how this mutation causes disease and affect the neurons in which way. And there, with these models, we could study the disease mechanism and also identify targets that can be used for drugs. But for Leigh syndrome, unfortunately, for mitochondrial disease, it's not easy or possible yet to generate animal models like mouse models or flies and so on. And for nuclear mutation, it is possible, but most of the time, a lot of models do not recapitulate what happens in the patients. So if we want to find drugs and we want to use a classical pipeline of drug discovery, one starts from target identification, high throughput screening, confirming the hit, and then finally working cells and preclinical models, and then going to humans. But this is not feasible so much for Leigh syndrome or other mitochondrial disease where these cells and animal models are not readily available. So this is challenging for Leigh syndrome. And so other opportunities needs to be found. And what we think is another strategy is to use induced pluripotent stem cells. So these are, you may heard of these. These are cells for which Shinya Yamanaka in Japan won the Nobel Prize for. And basically, the concept is that you can take cells from the body, you know, like skin cells, and you can reprogram them, so change their identity into with a cocktail of factors. You can change their identities from fibroblasts to pluripotent stem cells. That means they're pluripotent, means that they have the ability to generate all kinds of cells of the body, like neurons, muscles, liver cells. And they're also stem cells. That means that it can grow and proliferate and self-renew for a very long time. So they can be a very good source of material. So what can we do with this? So we can reprogram from patient into material into iPS cells, and then we can differentiate into neurons or brain organoids, what I showed you before, where we can study the mechanism of a disease in a dish. So basically that's a concept where you can start from somatic cells from a patient, reprogram them into iPS cells, and then differentiate them into specialized cells. And you can study disease modeling and function of the cells, and you can use this function to look for therapies and perform drug screenings. And you can also look at toxicity studies, and then with the idea of identifying new drugs that can be then suggested back for the patient. Of course, these cells are possible to be used for cellular cell therapy, so that you can bring the cells back to the patient, but this is not easily feasible. And for mitochondrial disease, where there is a lot of different cells that are affected. So, for example, in Parkinson, there's a possibility now they're starting a clinical trial on cellular replacement where neurons made from iPS cells and stem cells and can be implanted in the brain. But this is not yet possible for disease like Leigh syndrome, where we don't know yet if there's a specific area of neurons that are affected. But if you can replace them, then the function of the brain will be recovered. So these cellular therapy are not yet pursued. So what is instead done at the moment is, what we do in the lab is this disease modeling and drug discovery. So if you do drug discovery starting from iPS cells, this is how it looks like. So you immediately you can immediately use a material that comes from a patient, and it's a right, let's say, cellular type, for example, neurons, and then contain already the mutation from the patient or the features, and so one can identify a phenotype that can be used for screening, and then you are already in the relevant biological material, and potentially you can find a drug in a faster fashion. Of course, the challenge is that yeah, you can have effective. You need this could be representing an effective model system, and you can identify phenotypes, but you really need to be able to see these phenotypes. So phenotypes, I mean, you compare the cells, the neurons from the patient and the control, and you say, oh, this is different. For example, this calcium readout, these features of a membrane is different. Oh, this can be a phenotype, can be relevant for the progression of the disease. So I can use this as a screening for drugs. And this comes the second challenge, is to be able to have such a phenotype that is robust for screening, and also the cells they need to be grown in a very large number for screenings. So this is what we do. We started from mitochondrial patient. We isolate somatic fibroblast or blood cells. We reprogram to iPS cells. For nuclear DNA mutation, we can perform genome editing. For mitochondrial DNA mutation, this is not possible. I will not go into this. But once we have our lines of iPS cells, we can generate neuronal cell model like neurons, neuroprogenitor cells, which are the precursor of the neurons, or brain organoids. And these can be used to perform compound screening using a target or a phenotype that we see in vitro, with the hope to identify something that we can confirm in different models, finding a dose response, confirming toxicity, and then potentially suggest this in patients. So I'm going to give you two examples of this strategy. There were two works that we've done in the lab. So first is Leigh syndrome caused by SURFI mutations. And SURFI is a nuclear gene, so it is possible to generate mouse or animal models from this SURFI. But this model, although they show some mild deficiency in the activity of the mitochondria, they didn't show consistent neurological phenotype or neurological defects. And even the mice even show longer lifespan, so they live longer. So then they don't seem to be a very useful animal models to understand the disease. So the way we approach, we decided to use iPS cells to develop a new model. So we found that when we were after making iPS cells, try to make neurons, and we found we used the neurons that start from iPS cells, differentiate into NPCs, and then to the differentiated neurons 4 weeks and 8 weeks. And these neurons from the control, they are spiky, so they are showing activity, repetitive activity, but not from the patient. And also postsynaptic currents, which are a marker of how functional the neurons are. Also, these were not present in the SURFI neurons. So we found this defect in neurons, but we also found defect in brain organoids. So from iPS cells, again, we differentiate into ABs and then into brain organoids for until 90 days. And also here we saw that in the control, neurons are present and nicely organized in different layers, but this was not the case for patient SURFI organoids, that they show disorganized structure, even accumulation in areas where there should not be neurons and so on. So this is just a picture of how we make these organoids. You start from iPS cells and they start to aggregate, and then we embed them in a matrix that helps the organoids to be maintained in a certain shape, and then they grow over time and we can assess them. So we found that these organoids showed a disorganized structure because they were defective in their development. So they were less So they were smaller in size. So compared to the control organoids, the SURFI organoids were smaller. So here is the quantification. So the size was in the control was this, and in the patient was smaller. So and this is because the neuroprogenitor cells, so the precursor of the neurons, were not able to differentiate. So and they were not showing the correct organization. So in the control organoids, neuroprogenitor cells have a specific organization. So here are markers of neuroprogenitor cells here, and these are neurons. And here are neuroprogenitor cells. They show this sort of a rosette structure, these neurogenic zones, and this is lost in the SURFI organoid. So there are NPCs, neuroprogenitors, but they're not in the right organization. So what we think basically is that during the development of the neurons, there is a moment where NPCs or neuroprogenitor cells need to start relying on oxidative phosphorylation, so mitochondrial respiration, in order to make the neurons. But in the patient, when this is not possible, when NPCs, so neuroprogenitor cells, are not able to switch and use oxidative phosphorylation, because, for example, like in the case of SURFI or other mitochondrial disease, basically mitochondria are not functioning properly, so NPCs cannot start using oxals. They remain too proliferative and too unable to commit to neurons, and so they have a failure to commit, and then the neurons, they fail to mature, and they're smaller and they are less active. So we think that these NPCs might represent then a target of intervention. That means that we could try to see if we can improve this commitment of NPCs. So how can we do that? So in the papers that we just published, we found two potential strategies in respect to SURFI mutations. One is, so this is the scheme, so disease mechanism. So we think neuroprogenitors, they have an impaired commitment to OXPHOS, and so they proliferate too much and they don't differentiate enough. And that's why there is aberrant brain wiring, which could be also indicative of what we see in patients with this defective neuronal development. So we found as a strategy, one possibility to improve that is by using gene therapy, so gene augmentation therapy. That means you don't correct the mutation in the patient. So the patient still contains the mutation, but with viruses, so for example, AAV, which is mostly used for gene therapy, you provide a healthy copy of the gene, so that in a way, the mutated copies in the patient are still there, but you provide an additional healthy copy, and this helps the neuronal function by helping this commitment of the NPCs to neurons. Another strategy that we found is also to increase PGC1 alpha, which is basically a master regulator of mitochondrial biogenesis, which is the number of mitochondria. So by increasing the number of mitochondria, even if each of them still contain a mutation and still be a bit dysfunctional, altogether we might increase the energy output of the cells. As one example, we use bezafibrate for this. So we also tested antioxidant, but they didn't work in our model, so we didn't see any improvement of antioxidant. And also hypoxia, that was suggested as a therapy for mitochondrial disease and Leigh syndrome, also did not work in our case. But we found gene augmentation therapy and drugs improving the metabolic program like bezafibrate to work. So, so basically that's our concept. If you are able to activate in PGC1 alpha, then there's an increase in mitochondrial biogenesis, increase ATP generation, improve OXPHOS capacity, and so enhance neuronal commitment and maturation in NPCs. I'm going to give you the second example on ATP6 mutation, which is also these are also very common cause of Leigh syndrome, also of Mills, so maternally inherited Leigh syndrome. So ATP6 is a gene that is encoded by the mitochondrial genome. So for this, it's not possible to have animal models. This is the mitochondrial genome, and this is ATP6. So as you know, mitochondrial genome is present in multiple copies into each mitochondria. So if this is a mitochondria, I have multiple copies of the mitochondrial genome. And so this is the challenge. And also, you know, if you want to use to generate transgenic animals, like you can do for nuclear mutation, it's difficult because you need to have, you know, correct or introduce mutation in so many different copies. But also the other challenge is that the mitochondrial genome have different properties. There is no, so the repairing system is different. So CRISPR-Cas, for example, also would not work. And also because it's difficult to target RNA inside the mitochondria. So for all these reasons, it's challenging to edit mitochondrial genome. And so it's difficult to generate animal cellular models. So we use also iPSCs to generate a model system for ATP6 mutation. And basically, this is just a summary of this work. So basically, we use this mutation in 9185 in ATP6 gene. We had MPCs from patient, and we saw that also MPCs also in this case were dysfunctional. And they exhibited an increase in the mitochondrial memory potential, which caused a defective release of calcium. And so with that, neurons were less active and less functional. So we use this defect, this phenotype, this MMP defect for screening compounds. And we use FDA-approved drugs. So because we think that there are already a lot of drugs out there that maybe are used for another condition, or they're not used for any condition because they passed the phase 1 and they're safe, but they're not being, but then when they were trying to be assigned to a specific condition, let's say, I don't know, arthritis, then they were not effective, but still they're safe. So if we can use this pool of drugs and we can see if some of them does something in our system, then we can immediately suggest this to the patient. So we wouldn't need to do any more phase 1 or phase 2. We can immediately reposition these and the patient would have something that can be used immediately. So we found in that context of ATP6 mutation, we found as a potential drug savanafil, which was able to increase calcium in neurons and NPCs, increase their functionality. And now we're exploring, but savanafil is not, its use in children is not yet approved. So we're looking into drugs of this class to see whether something can be instead suggested. And we have some positive results that I cannot comment yet on, but it's very promising, and we're performing 4 compassionate treatments in different hospitals in Germany with very promising results. Hopefully I can present this data soon. So in conclusion, we think that iPSCs are a very good model system to develop neuronal models of Leigh syndrome due to nuclear mitochondrial mutations. We can use this system to generate neurons, brain organoids, and progenitor cells. We can understand the disease mechanism and where we think that neuroprogenitor cells are defective, and so their commitment to neurons is a key aspect here. We can use these models to identify targets of intervention, like the one I showed you, for example, PGC1 alpha or this savanafil. And then you can use this system to identify drugs that maybe are already FDA approved or safe drugs, and it can be repositioned for Leigh syndrome. But it might be that each genotype will require specific treatment. So for example, savanafil was not working for SURF1, and bezafibrate was not working for ATP6. So at this point, we're approaching in a way that we take one genotype and we study this separately. But we hope that over time, maybe we can identify common traits, common mechanisms that we could target. Yeah, so maybe there are common mechanisms that can be targeted. So with that, I conclude. So this is my team, and I would like to I would like to thank specifically Markus Schulke in Berlin, who is my collaboration partner. Especially, he's the one who is he's the physician who is taking care of finding the patient and now performing this compassionate treatment I mentioned. And so, yeah, so with that, I thank you for your attention, and I hope you, I was able to provide a useful view of what we're doing in the lab, and I hope that this technology may have even more and better ways to bring some potential therapies to the family that are desperately need some alternative for this disease like Leigh syndrome or other mitochondrial diseases that currently have no cures whatsoever. Thank you very much.